So good day. I am Eunice Bondad and together with my group mates, Nicole Chao, Dennis Javier, Aaron Lopez, and Kayla Publico, we're going to discuss the T and B lymphocyte development. So next slide please. So for the T cell development, first where is the actual T cell are being made? It comes from the red bone marrow where the actual T cell are being produced. We're gonna send the T cell to our primary lymphoid organ, which is the thymus gland, where it will mature and has a selective type of processes. It is good to take note that our primary lymphoid organ or the thymus gland allows the T cell development to to complete, and it is functional during the infancy, the childhood, and adolescence, because as we grow older, it undergoes atrophy where it becomes non-functional. T cell will go to our bloodstream and it will go to the thymus gland through the through the help of chemotaxis, where it will move from red bone marrow to thymus gland. And since our primary lymphoid organ is the thymus, our secondary lymphoid organs are the lymph nodes, spleen, and mucosa. So our T cell development starts from our stem cell to our lymphoid progenitor, to our double negative 1, double negative 2, double negative 3, and double negative 4 to our double positive. In our double positive, it is where our T cell will decide, or it will choose whether it will become helper T cell or cytotoxic T cell. In double positive, it is where our CD4 and CD8 will take place, because our cluster differentiator 4 and 8, they are the glycoprotein attached, which classically determine whether a T cell has developed um, T helper or T cytotoxic. Our helper T cells stop expressing our CD8, where it becomes CD8 negative, and it will continue to express CD4. Vice versa, our cytotoxic T cell will stop expressing CD4 and it will continue to express our CD8. Since they both stop expressing CD4 and 8, they are now called single positive cells. So, to continue with the discussion, Nicole Xiao will proceed.
Okay, so to continue, so the first marker that you're going to see is called the c-kit. So this c-kit, okay, it expresses from the stem cell stage up to the um, double, so you call this the double negative 2 stage. And then from there, we can see that CD44 expresses from the uh, donor negative 1 stage and the donor negative 2 stage, and it is the marker wherein you can classify this the cell that it is under the stage of double negative 1 or double negative 2. And then for CD marker 25, we have it is expressed under, or you can classify the cell if it is under double negative 2 or double negative 3 stage if the marker is CD25. So both CD44 and CD25 cannot be they cannot be expressed under double negative 4 stage. because they are only certain for double negative 1 up to double negative 3 stage. So if you can see here, so when call this when we are ano dito? So, tatagalogin ka na lang. Okay, para mabilis. So, for example, kapag sa isang laboratory, we are testing a cell. And then, yung cell ng patient na yun, nagpakita siya ng marker like CD44. Like, for example, kung negative yung CD44, tapos positive yung CD25, and then parehas nag-negative sa CD8 and sa CD4 na marker, it suggests that the cell or yung stage nung cell is nasa double negative 3 stage na siya kasi yung criteria niya, it simply expresses under the double negative 3 na stage. So yun, so all of these markers naman, they have their own function na ginagampanan. It's just that in this situation, the markers are simply for the classification of this cell kung nasa ang stage na ba sila. Ganun. And then, moving on, we have here the pre-TCR. So, yung pre-TCR naman natin, siya yung ang ina-express naman niya, kung makikita niyo sa picture, is sa double negative 3 stage siya and sa double negative 4 stage siya makikita. Yun yung, parang yun yung marker if nandun na sa stage na yun yung cells na yun. And then, later on, itong pre-TCR na to, magiging TCR siya. And this TCR receptor inaalaw na niya those T cells to identify which cells or which cells yung yung naapektuhan na nung pathogens from foreign pathogens. Ganun. And then this, so hindi siya masyadong kita sa picture, pero yung pre-TCR meron pa siyang another marker sa taas kasi pinagsamang pre-TCR and then yung CTR. So both that marker are accompanied by a marker called CD3 or other name niya is yung CD3 marker or CD3 T cell cofactor or co-receptor. I'm not sure, basta yun. Basta yun. Tapos this marker, it suggests na unlike sa T cell development and yung sa factor or sa pag-develop ng B cell, so mamaya yung B cell i-discuss siya. So parehas halos similar lang sila on the development, pero ang pinagkaiba lang is that instead of magkaroon ng large chain or large or heavy stain, not stain, I'm sorry, heavy chain doon sa T cell development natin, ang meron lang sa kanya is yung alpha chain and yung beta chains. So yun lang yung difference nilang dalawa dun sa B cell development. And then to further explain, here is Miss Javier.
So for the T cell receptor beta chain, this one, it is analogous to the B cell receptor heavy chain and it rearranges first. So yung rearrangement, it occurs between the double negative 2 bond and the double negative 3 cell. So once the beta chain is rearranged, it can be exposed by the double negative 3 cell, and this chain makes up the major component of the pre-T cell receptor. So the other less important component is called the pre-TCR alpha chain, but this should not be confused with the actual TCR alpha chain, which is recombined then expressed by double negative 4 cell and the double positive cell. So once the TCR alpha chain is expressed, it can combine with the TCR beta chain to form the T cell receptor. So, to discuss the formation of the B lymphocytes, here is Miss Aaron Lopez and Miss Kyla Publico.
The B cell development starts in the bone marrow. The bone marrow is the primary lymphoid organ where immune cells develop. In the early stage of B cell development, it takes place in the bone marrow. Then later, when B cells mature a little bit, it moves out to the secondary lymphoid organs such as lymph node and it develops there. Inside the bone marrow, there is an endosteal niche. An industrial niche is closer to the bones when the osteocytes are there, and there is another niche known as the vascular niche around the blood vessels. Next slide, please. Now, in the industrial niche, there are certain cells known as the stromatal cells, which plays a crucial role in B cell development. Next slide. The stromatal cells are actually supportive cells and they are large adherent cells. The Greek word stroma means matters. Stromatal cells work like a substratum which holds the pro-B cells in a specific niche, and it also influences their development by secreting several cytokines which are very important in B cells development. Next slide. Now, inside the industrial niche, there are hematopoietic pluripotent stem cells. And the hematopoietic stem cells can give rise to lymphoid progenitor cells, which is not shown here. Now, ultimately, this lymphoid progenitor would give rise to a pre-pro B cell, which is a total immature stage of a B cell. And at this stage, B cell express some adhesion molecules. Also, these pre-pro B cells expresses a B cell lineage marker, which is the B220. Also, at this stage, the B cell express another transcription factor, which is known as the EBF1, and we will learn about its function and its role in B cell development. Or next slide. At this point, the bone marrow stromatal cells express adhesion molecules known as the VCAM1. On the other hand, the pre-pro B cells expresses adhesion molecule known as the VLA4. Now, there will be an interaction between VLA4 and VCAM1, and it is very important for B cell development. VLA4 and VCAM1 interaction holds the pre-pro B cells near to this bone marrow stromatal cell, stromatal cell niche, such that the stromatal cell can influence its development. And also, this VLA4 and VCAM1 interaction gives rise to the expression of another adhesion molecule known as the c-KIT. And c-KIT has defined role from the pre-pro B cell after the interaction of VCAM1 and VLA4, and it starts expressing c-KIT. Next slide, please. On the other hand, the stromatal cell expresses stem cell factor or the SCF. This SCF and c-KIT interaction is important clue for the proliferative signal to B cell. And also another chemical known as the CXCL12 mediated signaling is very important for pre-pro B cell. Now, at this point of time, the two phenomena happens. One is c-KIT SCF interaction, which give rise to the proliferative signal, and also at this point, the DXL12-based signaling system give rise to proliferative signal also. From this point onwards, the heavy chain DJ combination starts. Next slide. Now, as time progresses, the only pro-B cell detaches from that specific stromatal cell and goes and bind to another stromatal cell, which are IL-7 secretive. Now, the IL-7 has distinct role in early pro-B cell development, and the IL-7 receptor is situated in the early pro-B cell. And the IL-7 also binds to IL-7 receptor, and this will give rise to specific signaling. At this point of time, the VDJ recombination takes place. So the B cell receptor, which is basically the membrane bound, and the BIGM or the anti-IgE, they are actually produced. The heavy chain is produced from this point onwards. So let's take a look on what IL-7 mediated signal do. Next slide, please. The IL-7 binds to its specific IL-7 receptor on the early pro-B stage, cell stage, and now it binds to the IL-7 receptor, which is a tyrosine kinase receptor. Now, along with it, Janus kinase is associated. The JAK gets phosphorylated upon IL-7 pointing, and it allows phosphorylation of the STAT5. To continue, here is Miss Lopez.
So the STAT5 moves to the nucleus and start transcription of NMYC and CMYC. Also, it starts transcribing EBF1. The EBF1 and STAT5 both allow transcription of PAX5. The EBF1 is super important for DGH recombination, and also PAX5 is very important for VDJ recombination. For the heavy chain recombination, PAX5 and EBF1 are super important. And these two transcription factors ensures that the heavy chain recombination takes place properly. Next slide. So now, the EBF1, the E2A, and the PAX5, all these transcription factors actually activate many genes which are important for the B cell lineage specification and B cell proliferation. Next slide. So once the early pro-B cell stage is over, then comes the late pro-B cell stage. In the late pro-B stage, it starts expressing a primitive version of BCR, which is known as B cell receptor. Next slide. Last slide. So at this point of time, you will see the heavy chain of the B cell receptor is formed, but the light chain at this point is not properly formed. And the light chain is known as the surrogate light chain, which comprise of V pre B or lambda 5. So this stage onwards, the light chain recombination would also start. Next slide. So now it would start expressing CD25 and high affinity IL-2 receptor, and it would become an immature B cell. Next. In the immature B cell, you can see the light chain is now formed, and also another remarkable thing is IL-7 receptor start regulated. So in the absence of IL-7 receptor mediated signaling, the adhesion molecules would be also down regulated. So these immature B cells are detached from the bone marrow stromal cell niche. Next slide. So now they come to the vascular niche, and they migrate via blood vessels and ultimately reach lymph node, which is a secondary lymphoid organ. Last slide. Pabalik pala. So inside the lymph node, there are specific regions for B cell and T cell development. So lymph node is like an army base camp where they get specific trainings. Now the B cell at this point is imagined. It's naive, though it can recognize antigen, but it has not encountered real antigen. But in this germinal center, in the specific B cell zone, it would interact with dendritic cells and other antigen projecting cells to recognize antigen. And it would either become an antibody producing plasma cell or it would become a memory B cell. So this is how B cell development progresses from the bone marrow and some part of the development happens in the lymph node. That's all for the B cell development. Thank you.
So that's all for the T and B lymphocyte development. We hope that you learned something today from us. That's all. Thank you po. So I will now stop the recording.